Hello boys, good morning to all of you, and this is meant for class 12 and we are here for providing a demo for you on experiment number 3, and this is for 9th January 2021. As we have been continuously giving demos week-wise, this week I am going to take another experiment, another interesting experiment. That is, and the aim of the experiment also is to prepare a temporary note of onion root tip to steady mitosis. That is the purpose. Purpose is to steady the mitosis. That's why you put the heading as study of mitosis or study of stages of mitosis. That is the aim. And whenever we want to perform an experiment, we require materials. What are the materials required? All are placed in front of you. Compound microscope, onion bulb, slides, cover slips, conical flask, needles, brush, forceps, and a chemical called acetylcholine. This one. This is acetylcholine. That is stain, dye. And the color of the dye, when we put the tips of the onion, it will be like this. Almost child color. Acetylcholine. Then petri dish, and of course water is essential. And another two chemicals which are very important is acetic acid and methanol. And this acetic acid and methanol are to be taken in 1:3 ratio. We have taken already acetic acid and methanol in 1:3 ratio. Okay? And then scissors, gas burner to heat up, to warm the roots, and the warming of the roots will be done in 1NHCl, hydrochloric acid. And the purpose of doing this is for hydrolysis, okay, to remove the water. Now, come to the third one, procedure. How to do, what to do. Already I said, procedure should be written in past tense because you are going to perform this experiment by yourself when you come back to school, and it should be pointless. Like I am giving a demo how to write. First one, fresh medium sized onion bulb is taken and old roots are trimmed off with the help of scissors. Now this is the onion bulb. Okay, the roots with the help of scissors trim off this, cut this roots like this. This is trimming. Okay, that is the first one that we need to do. Fresh, medium sized onion bulb is taken and old roots are trimmed off with the help of scissors. This is first point and the first activity. Second, a conical flask is taken with full of water. So here is the conical flask. Okay, with full of water. And place this onion bulb with the trimmed roots in this fashion, so that the left out roots will be in touch with the water. So onion bulb is placed on the conical flask in a reverse position with its base touching the water, and this setup must be kept for 2 to 3 days. This arrangement already our lab Kishore has kept already. This is kept for 2 to 3 days. That's how we obtain the roots. So for demo purpose I am telling you like this. Then fourth one, once the roots are grown after 2 to 3 days, we take the scissor, cut the roots. Now very important process in this experiment. If we don't do this activity correctly, the entire experiment will be a flop. So take care. Fixation is called. Fixation of the roots are done by placing the cut roots in a petri dish containing 1:3 acetic acid and methanol. So you have to take a petri dish. In the petri dish, you have to take that 1:3 ratio, 2 chemicals. One is acetic acid, 3 parts methanol. That's what we said. 1:3 acetic acid and methanol. You should take it. And put the roots in the mixture, and this makes the ongoing cell growth to be stopped. That's what we are saying, fixation. We fix the stages there. Whatever stage, like all of us know, of it, second chapter, then prophase, metaphase, anaphase, telophase. That's how one cell becomes two in all the bodies of animals as well as plants life. So we wanted to fix, because if the process goes on completely, we can't see what is the purpose of our experiment, to see various stages of mitosis. So we need to see all the 5 stages of mitosis. That is the purpose why we fix the root stages. Next, another important again, hydrolysis. This is done by warming root tips in 1 HCl. Now what we do, in another petri dish you take like this. In that, put 1 m normality, hydrochloric acid. Go to Bunsen burner, we have got gas burners in our lab. You go and just warm it. And once you warm it for how many minutes? 15 minutes. Don't boil it. The entire roots will be dissolved. Okay? Just for 15 minutes you do warming. And by doing this, be careful, you are in front of the gas burner. So one must be very careful with the fire. And then root tips are removed, wash with water. So take out the roots, transfer it to another petri dish, put water, wash them. Wash them means don't press them. Okay? And then now hydrolyzed root tips are placed in a drop of acetocarmine over a slide and cover slip is placed. Now once you wash that root tips with water, what is the next step? Take a cover slip. Take a slide. And on the slide, you put 1 drop of acetocarmine. Now we have got acetocarmine, we have got a ink pillow, just absorb it, put it on 1 drop on the slide. After putting it, what do? The hydrolyzed roots are there with you, ready-made, in the petri dish. So take out those roots with the help of forceps, place it on it. Now, after pressing it, put some water so that roots will not become dry, and then this is called cover slip, small thin glass. Okay. Now place this cover slip over the temporary mound. Again, I have told repeatedly last 2 times also that whenever you are putting cover slip, you should take the help of needle. So place the needle under the cover slip, right, and reverse the brush. Reverse the brush. Take the help of the reversal end and slowly, you need to have patience when you are doing experiment. So slowly you remove it. Okay? And while you are removing it, what is required? See that air bubbles will not be there. No air bubbles will be there under this. What happens if air bubbles are there? Those air bubbles will be like footballs and basketballs covering the tissue for which you are making this experiment. Now once the slide is ready, it's called now temper slide. Use the blotting paper to remove the excess stain and then place the temporary slide. And before you place it, one more important thing you need to do, that is take the needle, reverse it, or you can take the brush, reverse it, even if you can take pencil, reverse it, and tap it on the cover slip. But don't prove your stamina and strength. Slightly, just to tap it. Okay? Don't break it. So just tap it. What is the purpose? When you tap the tissue, the cells will be spread in your slide. That is the purpose. And once it is ready, place it under microscope, and on this I told you, what is that? Place it always under low power, 10 into 10, 100 times you are making it big. And now before you place it, the other things, formalities I told you, set the microscope. See that sufficient light is there. Okay? And then when you are rotating this object lens, I told you there will be touch sound. When you hear the touch sound, then only rotation is complete. Otherwise, blurred image, or sometimes you can't see the image. Right? And this is the eyepiece, as I have been telling to you. See through the eyepiece and just move the slide on the platform. At one place, you will see this, it is the procedure. Now, observation. What do you observe? As I said, in two ways you can observe. One is under low power, and once you are happy with the low power, now turn this disc, object disc. Okay, again you will hear that tuck sound. Okay, when you hear that tuck sound, means rotation is complete. And then after, once you place it under high power, do not operate this coarse adjustment. Always operate fine adjustment or smooth adjustment. Why? I told you the gap between the slide and the object length will be so narrow. You operate the coarse one, slide will be broken. So these are the precautions you need to take, and observe and see any stage. Again, for this to identify a stage, you require theoretical level. What are the characteristics of, let's say, I am picking up metaphase, for example, 1. Now what is that you will see? First, since it is a plant cell wall will be there, cell membrane will be there, nucleus you can't see. Okay? Nucleus is disappeared, nuclear membrane disappeared. You will see spindle fibers on both the north and south poles, and most important, cytoplasmic fibers or spindle fibers you will see. And what is the characteristic of this metaphase stage? How do you say that just by saying that the cell is in metaphase? If you happen to see short and stumpy chromosomes lying in the center of the plate, like as I am showing you in the chart, take it for granted it is metaphase. Meta means middle, phase is stage, middle stage. One character you pick up, like in prophase, you can pick up the homologous chromosomes, short, stumpy. In metaphase, you can see placement of all the chromosomes in the center. In the anaphase, you can see movement of chromosomes. You can see the movement of chromosomes north side and south pole. And of course, telophase, easily you can recognize if you find two nuclei. Normally, in normal cell, one nucleus will be there. A cell which is in telophase, there will be two nuclei. So this is how you need to pick up one or two characteristics of the stage and identify. Now, once you identify, we will give you a pointed eyepiece. So with the help of pointed eyepiece, you project which is the cell that is undergoing metaphase, and write why do you think that that cell is under metaphase. Right? 4 or 5 characters. A is there, B, C, D. Hence, the cell that I am observing is under metaphase, under anaphase, that is. Right? Now, come to the last one, conclusion or result. What is the conclusion? Always I told, your result or conclusion should be your aim. Have you achieved your aim? So, conclusion is, with the help of temporary mount of onion root prepared, mitosis stages are studied and understood. That should be your result. Next, precautions. First and foremost, onion bulb should be fresh. Second, I told you that when you are placing the onion bulb in the conical flask, it should be reversed. Second, third, the ratio, don't skip this, 1:3. Acetic acid methanol ratio should be same. And the fourth one I told you, when you are cutting the roots, it should be morning time, preferably morning time you cut the roots and fix it, then hydrolyze it. And of course, by preparing the slide, temporary slide, ensure that no air bubbles are there. So these are the things, and then draw at least one diagram. The stage that you are observing. Like I am observing my metaphors, so I have to label. Understand the significance of each label that you are taking, because external examiner will ask you questions on it. The moment you say it is metaphor, he will say, give me reason